So now I would like to introduce our guests for our second meeting. And I think it's very special because it's the last meeting of the year. We are near Christmas. So I thought that it would be very good to bring in Dr. Arturo, and he quickly agreed to be here. So thank you very much again for saying thank you a lot of times because I'm really grateful. So Dr. Arturo Casadevall, he's from John Hopkins Bloomberg School of Public Health. He has authored hundreds of influential scientific publications that I'm sure many of you know some of them, and also author of the book What If the Fungi Win and another book. But this one I thought that was special to bring here in the context of our meeting. The floor is yours. Thank you very much, Dr. Arturo. And then we can have questions after his presentation.
Thank you, Amanda. Can you hear me?
Yes.
So, go. So I thought, Amanda, sorry that I am hoarse. I have a cold. It's really freezing here in the United States, in Baltimore. So I'm pretty. Amanda asked me to talk about the book, What If Fungi Win? And what I thought I would do is just give you some highlights of the fungal kingdom. We're going to talk about the world's largest organisms or closest relatives, a cold kingdom, fungal melanins. We're going to talk about fungi and the origin of mammals, and fungi and global warming. So a little bit of each. Now, the world's largest organism is not a whale. It's this fungus, Armillaria ostoya. It lives in a Oregon forest here in North America. It's thought to be between 2,000 and 8,000 years old, and it's got the area of 3.5 square miles. That's probably about 6 square kilometers. And it's thought to weigh about 35,000 tons. So that's the weight of a battleship. And the reason that you know it's the same organism is because when you take up the mushrooms and you sequence them, you can see that they are the same and they're killing the trees above of it. Fungi are our closest relatives. So fungi are not closer to plants, they are in fact closer to animals. And this becomes very important because when it comes to looking for antifungal drugs, they're harder to find since metabolism is so similar. The kingdom is cold. This is some work that we did a few years ago by Radamés Cordero. If you take a thermal camera and you take pictures of mushrooms, you find that they are blue. And the reason for that is that they're colder than the environment. They maintain this cooling through evaporation. And as long as they have water, they remain cool. Now, what Radamés did is he also extended this to the microbial world. So when you plate Candida albicans, Cryptococcus uniformis, it turns out that the colonies are cooler than the agar. And what you see here on the right is, look at the diagram, you can see that going across a colony, the temperature of the agar rises because at the edge, you're having hydrolysis as the fungus tries to obtain food, and then it drops. The colonies are cooler than the agar, and so the fungi try to stay cooler. The mechanism is the same mechanism by which you feel cold when you take a bath. It's water evaporation. Fungi need a lot of water, and they use that water to stay cool. One of the fun aspects of this project is that we made an air conditioner based on fungi, and it actually dropped the temperature by about 10 degrees centigrade. Fungi are being used as substitutes for leather. This is an example of what can be done. Fungi can be grown into any shape, and then they can be baked to leather. And this does not require animals, and it feels the same. It's an example of what this kingdom can do for us. I'm going to spend a little bit time talking about melanin. The melanin is found in all fungi. The word comes from the Greek melan, or black, the pigment that is usually dark black to brown. It is found in all biological kingdom. It is made by probably all fungal species and is associated with numerous functions, but it is very resistant to acids, and that is how we isolate it. The world of melanins, you can see a picture here. The color of your skin is determined by the number of melanin granules. The ink of a squid is made out of melanin particles, and you can see bananas there trying to repair themselves by laying down melanin. So, fungi like Cryptococcus informans, if you give them the right substrate, turn completely black. And this melanin is deposited on the cell wall, and it's a very complex mechanism by which it is assembled. It's probably assembled in melanosomes inside the cell and then sent to the outside. Melanin can protect against many, many different things, from ultraviolet light to heat, to enzymatic degradation, to free radicals, defenses, heavy metals, and most importantly, it can protect against antifungal agents. So it's a non-specific type of armor. One interesting thing about is the insight that came from the Chernobyl disaster. Chernobyl was a chemical explosion in the old Soviet Union that it wasn't a nuclear explosion, but it led to a lot of contamination. So the site was closed off. Now, about 20 years ago, they sent in a robot into the area, and you could see the walls are all black. And it's black not because of the fire, but because they're growing with black mushrooms, black fungi. And one of the things we were curious of was whether the melanin was providing radiation protection. And the answer is yes. It provides about half the protection one would get from lead. But in fact, you can use melanin, to protect animals against lethal irradiation. Here is an experiment done in our lab in which you do black mushrooms, white mushrooms, or white mushrooms from synthetic melanin. And what you can see is that the mice will survive lethal irradiation. How does this happen? What happens is that the melanin protects the gut. And by protecting the gut, it allows the animals to recover their bone marrow. This has been confirmed by other people, and when we think that this may be useful in situations in which people are exposed to radiation. This is another group pretty much in India who showed the same result. So small amounts of melanin taken orally can protect the gut against radiation. Another fascinating aspect of this is that black fungi, grow towards radiation sources. This experiment done by Russian colleagues, and what you can see there is that the hypha, they move towards a radiation source. It's almost like they seek radiation. And some years ago, we were able to show that electromagnetic radiation can be used by melanin, can be captured by melanin to increase the growth of organisms. So fungi that are melanotic have some capacity to use radiation for food. And we were able to show that, in fact, when you irradiate melanin, it can convert NAD to an ADH. Now, we had a lot of trouble publishing this, but as you can see, when Nature and Science, everybody reviewed it, but nobody really wanted to take a risk on it. But when it was published, they had to write stories about it because it was so interesting. Now, one criticism of the work that bothered me was, X-rays go through you. Gamma rays are even more energetic. So the question be, how could melanin capture things like gamma rays? And Charles Turek at Savannah River Lab did the experiment that was definitive. What he showed was that if you made a melanin electrode, that the melanin electrode could generate electricity if put in a gamma ray beam, providing unequivocal evidence that melanin can interact with gamma rays. And this has now been confirmed by other groups. Now, today, melanotic fungi contaminate the space station. And in fact, people have documented that melanotic fungi grow much faster in space. And we think that is the result of the radiation environment. However, fungi can also be used to help us colonize the world. So NASA has a big project, what is called the mycologist, in which they're trying to understand how to use fungi to help us survive space travel. If you look at the microbial pigments, if you look at the black mantis of Antarctica, that's the black comes from a lichen. That's a symbiosis between an alga and a fungus. The fungus is black. What we think is that these are being used for radiation capture, solar capture. So I want to introduce you to a new concept, and that is the concept of disaster microbiology. So whenever there's a disaster, we all become aware of it. The houses are destroyed, the people, are killed. But what people don't realize is that the microbes are also altered. Wherever you have a tsunami, a radiation, a forest fire, it tends to affect all the microbes in the area. So the Earth is undergoing disastrous global warming, and temperature is rising rapidly. And this is going to affect all the microbes because they can either have to adapt or die. Now, I will tell you how dangerous this is by showing you the Permian extinction event. So 255 million years ago, 90% of all life on Earth was wiped out. It was the greatest extinction in geologic history. The current understanding is that it occurred over 80,000 years and was associated with climate change. That resulted in increased CO2 and reduced oxygen. It came from a fire where there is now Siberia. There was a huge coal fire that burned for almost 100,000 years. And this coal fire generated a lot of CO2 and acid rain and warmed the planet. The fungi, however, do very well in this situation. This is a picture from a mountain in South Africa, and the Permian is separated from the early Triassic by 1 meter of fungal spores. So think about it. This entire ecosystem died, but the fungi proliferated, and today you can find the fungal fossils from that time. And they look like plant pathogenic fungi. So the importance of this is that anything that survived the calamity would have had to deal with a fungal world. Fungi may have been selecting who survives into the Triassic. The fungal kingdom is thought to be the largest kingdom, maybe more than 6 million species. It includes major pathogens of plants, insects, invertebrates, and ectothermic vertebrates. Those are cold-blooded vertebrates. They're currently devastating major ecosystems. For example, the bats in North America. You have a catastrophic amphibian declines from a chytrid fungus. Salamanders are being killed from another chytrid fungus, snakes in North America. But yet mammals are remarkably resilient. In fact, we don't tend to worry about dying of fungal disease. We worry about bacterial diseases, viruses, parasites, but most people within that community don't worry about the fungi. On top of that, there are very few fungal species that are pathogenic for humans. In fact, about 10. If you know 10, you can you can think of more than 90% of the fungal species, the important ones. And they tend to come from hosts, like Candida albicans comes from your mother, whoever took care of you as a baby. Dermatophytes are everywhere, or they come from the environment. And these are well-known fungal pathogens. In Brazil, you can add to this paracoxy. And so the question is, why mammals? Why are we the dominant large animals in the world today? When you go outside, what do you see? You see mammals and you see birds. You may see reptiles, but they tend to be small, few, and they tend to be smaller and hidden. The origin of mammals go back to the Permian. But they don't really take off as a major group of organisms till the late Cretaceous, about 65 million years ago. So one of the questions that have fascinated me is, why mammals? How do you select for a group of organisms that requires so much food? We eat 3 to 4 times a day, and most of the calories go to maintain temperature. Well, high temperature gives you a stable internal environment. It gives you the capacity to move and digest food in cold weather. But I would also argue it gives you great protection against infectious diseases. To put it in perspective, the plot at the left shows the energy required of mammals. So us elephants need about 1,000 times more energy to get through the day than a frog, 10,000 times more than a fly. And this is normalizing per kilogram. It's a very, very energy intensive lifestyle. And this lifestyle takes off after the end of the Cretaceous. We're going to talk more about that. But what you see is this great mammalian radiation, in which is giving us everything from humans to whales to bats. And I'm very interested on how do you select for a group of organisms that need so much food. So today, if you're outside, you see birds and mammals, and they both tend to be warmer than the environment. What is the evidence that endothermy protects against fungal diseases? This is an experiment done by Daniel Smith, who's a postdoc in our laboratory. He went collecting fungi here in Baltimore and plated them. And what you can see there is that the environmental fungi grow without any problem at 30 degrees. But only one grew at 42 degrees, and that was Candida tropicalis, which, as you know, is a human pathogen. Today, ectothermic organisms, cold-blooded, are vulnerable to fungal diseases. For example, the corals, they get damaged by global warming, and then fungi work on them. Frogs, salamanders are very vulnerable. Turtles are being killed by fusarium. The fusarium goes right through the shell. And in North America, we're having snakes die off. What the common denominator, and also everybody knows that they're major pathogen plants. The common denominator is all these species don't regulate their own temperature. Evidence for the power of temperature comes from this paper. It shows that you can take frogs, and if you create warm cinder blocks, the frogs will climb on them and cure themselves of chytrid. They know to seek hot environments. Fungal diseases in humans are more common in the cooler parts of the body. Nail fungus, athlete's foot, dandruff, which is a fungal infection called malacisia, and in babies, diaper rash. These tend to occur away from the core. The core is 37 degrees. The temperature in the skin is about 30 degrees. John Perfect and others have carried out experiments that show this experimentally. You take a rabbit, and you cannot give a systemic infection with Cryptococcus. It simply clears it. But you can give a skin infection or infection on the testicles, which are cooler. But if you give a systemic infection, and steroids, you get a dead rabbit. So the bottom line is you seem to need both adaptive immunity and endothermy. And this is why we tend not to have to worry about fungal diseases. The white nose syndrome in bats uh, emerged in the United States in 2006, is now spread throughout all of North America. Basically, the bats are fine in the summer when their temperature is 37. However, to survive the winter, they hibernate and their temperature drops to 10 to 12 degrees. When that happens, a fungus grows on them and kills them. If you take the infected bats into a lab and you feed them and raise their temperature, they're able to clear the infection, giving you a dramatic demonstration of how temperature protects us. Some years ago, we analyzed the temperature tolerance of fungal species. This was done with Vincent Robert, and we looked at all the maximal temperatures in a collection in the Netherlands. And what you find is that the fungi do very well up to 30 degrees, but after 30 degrees, most species decline rapidly, such that for every degree that you can go above 30, you can exclude 6% of fungal species. So why are we 37? Well, Aviv Bergman gave us a potential explanation. He solved two simultaneous equations. One is the benefit that you get from temperature on protection against the fungi, and the other one is how much energy you need to eat. And he found that the optima was our temperature, which was about 37 degrees. So we seem to, something in the past selected for our temperature. And it was to be a temperature where we didn't have to eat all the time. You can be hotter, but you will have to eat a lot more. So what could that have been? Well, when you look at history, geologic history, you can see it's the age of the dinosaurs up till the end of the Cretaceous. Then you have what is called the Tertiary or the Paleogene, and then it's the age of mammals. So the dinosaurs died out, and the mammals proliferate. And it turns out that there was massive fungal proliferation also at the KT boundary. So people ask, what killed the dinosaurs? I think it depends who you ask. It used to be that people thought that small mammals ate the eggs. That doesn't make any sense. This doesn't make any sense either. They didn't die from smoking, but I found this on the internet, so I share it with you. Massive volcanism could have really contributed. This is in the area of Deca today. There are remains of volcanoes that opened up 65 million years ago, and they could have had major effects on the climate. Most people now associate the end of the dinosaurs with an asteroid that hit the Yucatan. This would have caused a tremendous global calamity and probably killed the dinosaurs from the blast effects. There are two other explanations out there. This, believe it or not, was published in Current Biology, which is a cell journal. Some people speculated that the dinosaurs may have passed enough gas to have triggered global warming. I don't believe that, but it's out there in the literature, and it's fun to think about. And the next explanation, I'll give it to you in silence because it's not a scientific explanation. So with that, let's talk about the Cretaceous world on its end. The Cretaceous, it was a warm, volcanic, and forested planet. The Atlantic was very small. The continents were separating, and the world was dominated by a reptilian megafauna. And the impact of the occur in the Yucatan, where today the ring of the Chicxulub crater has been found. The post-impact world was completely devastated. You had all the forests come down. And then you had fires because you had a lot of material ejected into low orbit that then re-entered as flaming meteorites. Temperatures dropped in one day or about 10 to 12 degrees centigrade. because the fires, the smoke obscure the sun. And in this world, you had fungal proliferation. This is documented in the geologic record. So imagine the vegetations die off, the fungi proliferate. As the fungi proliferate, you have a lot more spores. And these spores could have selected for organisms, animals, that were more resistant to fungal disease. And that would have been the early mammals. Here's how you see it. There's a fungal spike at the Cretaceous. So the fungi separate. On the top part, you see conifers and flowering plants. And then you see the calamity, which is highlighted by an increase in iridium. The iridium came from the asteroid and it spread throughout the world. And then you have no more plants, and then the fungi proliferate, and eventually the ferns return. So in the post-impact world, the large animals would have been killed by the blast, the disruption of food sources, the chilling of the planet, maybe fungal diseases. Reptiles fight off fungal infections with induced fevers, but there was no sun. Reptilian eggs are susceptible to fungal diseases. In fact, today you can find dinosaur eggs already with hypha growing in them. So just like today, the fungi kill the fusarium kills the turtles. Back then, fungi probably killed dinosaur eggs. So I proposed that a fungal filter at the end of the Cretaceous selects for endothermy in mammals. And this has been written. Some of the papers are being published, and it's a very simple idea. Once the calamity happened, you had survivors. You had small reptiles and small mammals. But then the mammals got selected. They may have been selected by the fungal bloom. They may have been selected by the fact that they could move around in a colder world. And when the skies cleared and the planet recovered, there were just a lot more mammals than there were reptiles. And the net result is that you usher in the age of mammals, the group. So this actually was written, first time I wrote this was 20 years ago, and I have updated. Since then, I updated whenever I learn more about things and bring in additional information. So the fungal infection mammalian selection theory has a lot of explanatory power. By the way, I never had pushback. It could be that nobody cares. But to me, it helps explain something that doesn't make a lot of sense, which is how do we end up with all the large animals being mammals that need so much food? It provides a selective mechanism for an energetically costly lifestyle. It explains why most mammalian temperatures are in the high 30s. It explains the tremendous resistance of mammals to fungal disease. It explains it poses endothermy as a major host defense mechanism, and it explains the minor role for fever today. Why? Well, think about it. Today, when people get an infection, doctors immediately give analgesics not analgesics, antipyretics to lower the temperature. The reason is that most diseases that we get infected with today are already temperature adapted. But if you go out into the countryside and you fall and you get a gash in your leg with dirt, having those a couple of degrees could provide tremendous protection against environmental organisms. So now we're dealing with a warmer world. And we're going to have to worry about fungal adaptation to temperature. So today, most pathogenic fungal species reside near the equator. That kind of makes sense. To cause disease, you need to have both virulent factors and temperature adaptability. And it stands to reason that organisms near the equator would be more likely to grow at higher temperatures. Fungi, however, can be rapidly evolved for thermotolerance. This is a paper that evolved a insect-killing fungus to grow at 37 degrees over a period of a few months. Why did they do this? They did this because once the insects get infected, they sun themselves and they kill the fungus. So by making the fungus more thermotolerant, they were trying to make a better insecticide that would not require chemicals. Cities are heat islands. Cities are considerably warmer than the environment by about 4 to 5 degrees centigrade. There have been already a couple of studies that have shown that when you compare the fungi from cities to the fungi from the environment, from the countryside, those from the cities are better adapted to higher temperature. And we decided to do this here in Baltimore. Baltimore is a heat island. This is what our harbor looks like. And the number of hot days is going up significantly every year. So Daniel Smith went to a cold neighborhood, cooler neighborhood, and a warmer neighborhood. The cooler neighborhood has a lot of trees. It's a couple of degrees centigrade cooler than the hotter neighborhood. And he looked at the fungal species. So he asked the following questions. Are urban fungi more temperature resistant than expected? Are fungi in warmer neighborhoods more thermotolerant than those in cooler neighborhoods? And what fungi are present in the warmer areas? Are they different? Well, so he found significant differences. One of the differences is that in the warmer neighborhoods, the fungi are losing their melanin. And the reason for that is so that they don't overheat in the summer. They're more white than the ones in the cooler neighborhood. And he also found that when he did heat ramp experiments, that means that you put the fungus in a PCR machine, really, and you turn up the heat, that the ones from the warmer neighborhood was more adapted to higher temperature. So what you're seeing here is within one city, within about 5 kilometers, 2 neighborhoods differ in temperature, and the fungi from the warmer neighborhood are more temperature adapted. So this is an example of what I worried about 15 years ago, in which we argued that global warming will bring new fungal diseases from animals. And what I'm going to argue now is that this may have already happened with Candida auris. Candida auris was first isolated from the ear canal of a patient in 2007, hence the name auris, which means ear in Latin. So people have looked at collections to see if it was missed, and they found a couple of isolates. But this is clearly a new pathogenic fungus. And here is what's difficult to explain. Between 2009 and 2013, you had nearly simultaneous emergence in 3 continents. So this happened in South America, Asia, and Africa. And the isolates are not related. It's not like somebody took an airplane and brought them away. So I ask you, how did you get a new pathogenic fungus emerging independently in 3 continents? What will be the common denominator? Well, there are 3 theories. One of them was there all the time, but people missed it. The problem is that when people went back into collections, it was not there. There were an occasional isolate, but it was not there. This is a new fungus, the medicine. It was a consequence of overuse of azoles in medicine and agriculture. The problem is, it's unclear how antibiotic selection will give you a new virulence species. And we propose that it's the result of climate warming, which selected for variants that could grow at human temperatures. So the problem here is that this is very difficult to prove. It's conjecture, and you're going to have to look at circumstantial evidence. So one form of circumstantial evidence is to analyze the temperature susceptibility of close relatives. And when you do that, you find that the C. auris appears to have adapted rapidly, whereas its neighbors have not. It's now been isolated in the environment. It's been isolated by Dr. Chaudhry in India from the Andaman Islands. And she found it in 2 places. She found it in a beach that was frequented by people who may have put it there, and from a very remote marsh. The people don't go into the remote marsh. It turns out that the isolate from the remote marsh had trouble growing at 37 degrees. Eventually grew, but it gives you a sense that maybe the wild organisms are adapting, but many of them are not fully temperature adapted yet. Now, to make matters worse, people are getting colder. Yes, your temperature is about a degree colder than your great grandparents. How could this be? Well, it turns out that in the last 100 years, temperatures have been dropping for both men and women. And what is thought to have happened is that we live in a cleaner world with less disease. Back then, a lot of people had worms, they had indolent tuberculosis, and they had more inflammation. And inflammation generates heat. So as we move into a cleaner world, a world with less infectious disease, our temperature has dropped. But you see the problem. If the fungi are adapting and our temperatures are coming down, there may be a collision soon. To add to the problem, mutagenesis increases with temperature. This is very nice work done by Dr. Gusa at Duke that showed that transposons are activated by temperature. And so what happens is that as you go up in temperature, you get mutagenesis. This will mean more variants. The majority of mutants are less fit, but occasional ones have enhanced virulence and lower antifungal susceptibility. So you no longer have adaptation, but you have adaptation and mutation. And this could give us problems that are unknown to us. I'm near the end, and I thought some of you may be familiar with this story, The Last of Us. When this came out here in the United States, I get a lot of phone calls from journalists asking me, were you an advisor to this? I said, I never heard of it. But the idea is the same. Yet we know that cordyceps can turn ants into zombies. They, in fact, replace the ant nervous system and make the ant crawl up to the top of a tree. Could something like that happen to people? Well, my answer is always the same. Improbable, but not impossible. Improbable because I don't know of any fungus that can turn a mammal into a zombie. But it's not impossible because we know that fungi make things like LSD. They make compounds that can give you hallucinations. So is there a fungus out there that if it gets into people can affect them to behave like zombies? I don't think so, but I don't really know. So with that, I think I lost my last slide. I lost my last slide. Yep, I lost my last slide, but it's all right. It was just a summary of everything. And we finished on time, so that I'm happy to take questions and discuss anything you want. And I'm really sorry for my voice. I think that this is a successful talk because I have not lost my voice, which was one of my
Thank you
fears.
very much.
Thank you,
Thank you
Amanda.
very much, Dr. Arturo. And sorry for you be sick and thank you very much even more because you were here even sick. So for us that's not from the US is also good because you can speak low, so it's easier for you to understand even better the voice like that. So we already have questions in the chat, and also I'm sure people here also have questions. But I think I will start with the questions so people also can feel more comfortable. If you have question, you turn on your camera and your microphone and you can speak. But reading for your book and also seeing for your talk, I'm so impressed how you know a lot of different things and put all together in a great hypothesis that things make sense. And sometimes nowadays we study and we got so much thinking about what we are specialists, and then we lost to learn different things that could help us in our knowledge and our theory and hypothesis. And then reading our book, I saw that your life story, you have studied something related to chemistry before go to medicine, and you had great experience from that. And then when you writing to the young scientists, you say that you should be doing different things as well, thinking out the box and to be very prepared because we are seeing probably you are correct. So you are in big trouble because you are the next scientist handle with this global warming resistant fungi in the future. So I like to hear from you. What do you think that us as young scientists
Well,
could be?
what I would tell all of you is that I could not have given a talk like this when I was a graduate student. Okay, I just couldn't do it. Part of the reason is that I needed to learn a lot of stuff that you can put together to talk into things differently. The problem is that our educations are very specialized, right? We basically think about it. You go to a PhD program and they tell you learn more about less and less, become specialized, focus, focus, focus. But that's not the reason I went into science. I went into science because I was curious. I'm interested in a lot of things. And what I have done, and if you can do it on your own, is you can train yourself to be a generalist by yourself. No one can do it for you because your own teachers and mentors are specialists. So how do you do it? This is my advice to you. How much I ask everybody, don't have to answer. I know nobody can answer. How much time you spend on your phone each day? Imagine you just take 5 minutes each day and look up something related to science in other fields. And the best way to get hints of what to look at is look at the science news. When you hear something about astronomy, something about oceanography, try to read a little bit more about it. And what you find is that if you do that, you're going to begin to acquire a much bigger breadth of it. You don't have to know quantum mechanics to know, for example, that they did some experiment that show a parity violation. You could just say, why is this important? And try to get a sense of it. And what you do is, and then you begin to make good new ideas. And this will give you, excuse me, this will give you a whole new set of tools. And it's a lot of fun. And life becomes more interesting. Life becomes better because you're engaging on a lot of things that you don't ordinarily engage in. But you have to do it yourself. And I promise you, no more than 5 minutes per day.
This is good. Anyone else has a question? Thank you very much. Meanwhile, I can read one from the chat. Professor Casadevall, it is always a pleasure to listen to you. Okay. In terms of virulence, do you think that melanization in pathogens such as Cryptococcus could have evolved primarily as an adaptation to ancient environmental radiation rather than as a response to mammalian immune pressures?
Yes, I think melanin was here long before there were animals. Melanin is an ancient pigment, and you know that it's ancient because you can find it in all life forms. So it was there from the beginning. This is a pigment that protects cells. It was never put there to cause disease, to be a virulence factor. It functions as a virulence factor by virtue of the properties that are selected for the environment. It helps protect against metals in the environment. It helps protect against free radicals. So when a fungus meets a melanized fungus meets a cell, for example, that puts an oxidative burst, the melanin can help. So what appears to be a great virulence factor in reality is a pigment that was just selected for survival in the environment.
Another question is, could current environmental warming select for greater thermal tolerance in Candida auris, allowing it to overcome additional thermal barriers in mammals and expand its pathogenic potential in other mammals?
Yes, I think it could, although I think it's already there because the recent clinical isolates can grow at 42 degrees. At 42 degrees. It's interesting when you look back at Candida auris about 10 or 15 years ago, many of the cases were skin infections. So it's gotten into humans, is evolving with humans, but now the isolates that you recover are very much temperature adapted. So, I don't know. I guess rabbits have a higher temperature than we do. They're 40 to 41 degrees centigrade. I suspect that Candida auris can now cause disease in them too.
Anyone else has any question? Carlos?
Thank you. Hi, everyone. Dr. Casadevall,
Hi,
it's
Carlos.
a pleasure that I can hear. I'm from Mexico, and I have some question for you. Um, in that case that specifically talk about Aspergillus, um, like at the present has no pathogen opportunistic with a several characteristic of factor of virulence, like the melanin, the temperature, and another that uh, you study in the past. In the future, we can talk about Aspergillus like a, a la pathogen primary.
Yeah, I mean, I think that Aspergillus is another one of these organisms that it doesn't cause invasive disease if you have intact immunity. Aspergillus has no interest in causing disease in mammals. All these virulent factors were selected for survival in the environment. And I think that one of the important things for people who work on fungi is to do that thinking and saying, okay, I'm working on this virulence factor, and I know that it helps the fungus survive against the immune system. But what was their function in the environment? And that could give you new insights on how the evolution of virulence.
Thank you, doctor.
Thank you, Carlos.
We have only more 7 minutes, so if you want questions, the time is now. For now, I also want to talk about. I work with sport trichosis here in Brazil. That's epidemic because there's zoonotic
Right.
transmission in the cats. And then even my paper CTR hypothesis for Candida emergence because we see that it also independent emergence in different states of Brazil, and all of them were capable to infect cat and then transmitted and causing outbreaks. So we
So
also
Amanda,
think,
that is that this Sporotrix epidemic in Brazil, as you know, people have argued that it's probably global warming.
yes,
And I think that there are some things you could do. It'd be interesting to check the thermal susceptibility of the clinical isolates versus earlier isolates, because sporotrichosis only gave you skin infections, right? It's seldom
yes,
disseminated. So if you collect a bunch of isolates from cats and you collect some of the ones who are known from the past collections or something, is there a difference in temperature?
it's a good question. I know that they compared the temperature survival in different isolates, and some strains are more resistant than others, but I didn't see any paper that they compare the timing of collection. It's a good idea.
To
And
look over
we know
time.
that yeah, it's a good idea. When I have my own research group, maybe I'll focus on this.
It's also worthwhile
It's
if
good.
one could get some environmental isolates. It may be just like the Candida auris, that they're just not very temperature adapted yet.
Yeah,
I think it's
it's
a very
interesting.
interesting idea, and I think that if I was in Brazil, I would work to try to make the association between climate change and the new outbreaks. For
Thank you,
example,
Arthur.
is it more common in parts of the country that are warmer?
So this is like waiting discussion as well, because we are seeing new strains emerging in Chile in a colder place, when it's even colder than like the south, and we are very curious about that as well.
But the colder parts of the planet are warming faster. So in the case of Chile, it'd be interesting to know what's happened to that area relative to the way that it was in the past.
Yeah, we have here Eileen from Chile that's working with politics in Chile. I don't know if you want to mention something like about this, Eileen. Well, she's still in the meeting, but yeah, it would be interesting to investigate. Yeah,
And we have a question from Jose.
We have another question from Brunelli, from Dr. Brunelli.
Can you read, Ramesh?
Yes, of course. Dr. Brunelli is asking, could current environmental warming select for greater thermal tolerance in Candida auris, allowing it to overcome additional thermal barriers in mammals and expand its pathogenic potential in other mammals? Also, he has another question, but I can read
Let me
it.
answer that. It may, for example, begin to turn up in birds, because birds are associated with some of these Candida species, but they don't cause disease. The birds are pretty hot. But if it adapts further, we may be getting to see birds coming down with Candida auris. And the other one says, how strong is the current evidence that global warming is expanding the ecological niche of coccidioides beyond traditional endemic areas, and what could be done to address this? So the evidence is pretty strong. Here in the United States, many of the cases are now occurring outside of the old geographic boundaries, and people don't have travel history. So it's happening. What could be done to address this? I'm not sure anything can be done, because unless we can work on the climate. But I think the important thing is the physicians need to know. There are sites in South America of coccidiomycosis. One question may be that you also begin to see spread outside of those regions.
We have fewer cases demonstrated by presence of antibody in Venezuela by serology of coccidioides outside its traditional restricted area.
Hi, Dr. Casadevall. I have a question as well. So I just finished up my PhD. I spent about 3 years studying Candida auris, and to really summarize my work, I discovered, and I think you may have seen my paper, it's under revisions, that lipid synthesis is something that seems to be key for Candida auris to persist in a skin-like model. So obviously the main classes of antifungals target the plasma membrane, and I'm just wondering if you think that lipid synthesis itself might be something that could be targeted to treat Candida infections, and not just Candida auris, but maybe the other Candida species that it's closely related to. Thank you.
Absolutely. I mean, the key is to find a difference in metabolism between lipid synthesis in Candida and lipid synthesis in humans. Because the problem is that, as you know, many of those enzymes are the same. So you got to find some difference that is druggable. But there is something for you to think about. When a fungus infects a person from the environment, it must undergo a tremendous temperature increase, right? You're going to go from the ambient temperature, and we may be able, if we know what a fungus does to adapt to higher temperature, it may be possible to target that with drugs. Because if you can give the human a drug that